Thanks for joining another episode of Imperfect but Improving, where we progress our understanding of the world by speaking to the experts that are progressing the frontier of our scientific understanding. I'm so excited for today's conversation with Clivia Lasovsky, Martin Wikelski, and Christian Kurtz, who have a new paper out in Science, one of the most reputable journals in all of academia, focusing on how pigeons have their magnificent ability of magnetoreception. Thank you all for taking the time to talk to me. I really am so excited about this.
Pleasure to be here.
All right. So jumping right in, I want to start at the top by asking, how do we know that pigeons and other birds have this ability of magnetoreception, this ability to sense Earth's magnetic fields in the first place?
I think the idea came about by the fact that a lot of the songbirds migrate at night over long distances and they keep a direction. And I think the best data we have on that is actually from Bill Cochran out of the Midwest in the 70s and 80s, where he built transmission devices, little radios, little transmitters, followed individuals and especially one individual for 6 nights and showed that the magnetic direction that it follows is actually always the same. It's the same angle, and it's the same angle to the sun. So it changes a little bit from night to night. And then the question was, is that real? And what we did then with Bill Cochran was to change the magnetic field, twist it by 90 degrees. And then the birds at night in clouds, in complete darkness, actually went 90 degrees west. So off the regular track. And then the next night they went again 90 degrees to the north. So that means you can really show that they are following a magnetic field. You can fool the magnetic field by sort of calibrating it at sunset in the wrong direction. And that really showed that they are using that. There are other experiments in lab settings that show similar systems, but in lab settings, you never know for sure what that means. So therefore, I think the experiments in the wild are the key to really understand that they, at least sometimes, use a magnetic direction system.
And so the evidence is really that by applying another external magnetic field, we show that their navigational abilities are messed and no longer as good as they were. And that's how we eliminate the fact that, they're not just using landmarks or other visual cues to navigate in these situations.
Yes, or an inertial system that is sort of a gyroscope and different instruments that are keeping you exactly on track. But that's impossible in a bird, and therefore the only option at night in clouds where you have no other visual cues is a magnetic direction.
Okay, wonderful. And I'll ask this now just so we can circle back to it later. Do we have an estimate of how widespread this ability is across migratory birds?
We don't know, but just by default, they all seem to do the same thing. And therefore, everybody assumes that they all have that same system. One other aspect that I think is absolutely critical is that people always mix up the question of a magnetic map and a magnetic direction, because the magnetic direction is what we showed in the wild and it really exists and you can screw with it, the direction finding system. But there are people that think that you can navigate by a magnetic map. So you have sort of crossing magnetic lines that you can determine where on Earth you are. And for me, that doesn't exist. I don't see the evidence. It's not shown in the wild, and therefore magnetic direction finding, yes. Magnetic map, no.
So just to make sure I'm understanding this, that is to say that you believe there's evidence that birds can use the magnetic direction as a compass in order to figure out, okay, I need to fly, 30 degrees northeast or whatever. But given that they're transported to a specific location, they can't coordinate their longitude, latitude precisely.
Exactly.
Part of my fascination with this was reading about the puzzle that this presents as far as the Earth's magnetic field is quite weak in any given biological system compared to the thermal noise of its molecules jostling about. And so it's kind of a puzzle how biology could have come up with a mechanism to detect such a weak force amidst the chaos of warm biological systems. Could you go through what have been some of the leading explanations behind this ability of magnetoreception?
Yeah, basically there are, let's say, two main directions. So one part of the scientists believe that there are some kind of cryptochromes, which are like light-sensitive, UVA, light-sensitive proteins in the eye of birds. And it is believed that these cryptochromes, so they sense a lot. And then supposedly this leads to a conformational change, to some changes in gene expression. And then this change in gene expression would somehow be transmitted to the brain of the birds. That is one idea. And the second idea is more magnetite-based. So here it is also known that little magnetite particles are existent in several organs, in the brain, in all kinds of cellular structures, endosomes, lysosomes, in various cells. And then the idea is while, for example, the bird is moving through the magnetic field, these magnetite particles would, like a microscopic compass needle, align to this magnetic field. And this would also lead to an intracellular signaling pathway. And then via the nerve system, this information would be transmitted to the brain and lead to a reaction by changing flight direction. So these are the two fields that are kind of existing. And we know cryptochromes exist and we know magnetite particles exist. Which one of these would be the most relevant for magnetoreception? This is still highly debated, as Martin just said. So there's still a lot of theories and hypotheses out there that are debated.
And crucially, as I understand it, no experiment has been done showing that if we intervene with either of these methods, then the birds lose their ability to sense the Earth's magnetic fields and navigate by them. Is that correct?
Yeah, that's correct. Because it's very difficult. How would you, deplete or knock out cryptochromes? So that's not possible. And it is very difficult to find methods to investigate this in a natural environment, as Martin just explained. You can put the birds into some magnetic cage, but this is very artificial and it doesn't resemble what is outside in nature. And with our paper now, that since we targeted the macrophages and showed that in a natural environment, when we deplete the macrophages, the birds were unable to find their home loft. This is actually a very new method, and with a new method, then it also reveals new results.
It's such an intriguing finding. And before we get into that in depth, Martin, Christian, I want to ask about what was the genesis of this experiment. What led to the hypothesis that this is something worthwhile to look into?
This was just serendipity. Martin and I, we met at a conference, and I was talking to Martin that we had just found these stupid and silly magnetic macrophages. We use mice as animal models, and these magnetic cells, they interfere with one of the most widely used cell enrichment methods. So when you have a mixture of different immune cells and you want to take out some that you're interested in, then you can label them with an antibody, with a specific antibody that has a small magnet. And then you can pass it over a magnetic column. And these cells would be would get stuck. The others pass through the column, and then you switch off the magnet, and you can elude the cell of interest and study it. And we found that there were always some contaminating macrophages that stuck to the column, although they did not express the receptor for this antibody. So, and it was really not dependent on the antibody. They also would stick to the column without any antibody added. So they must be intrinsically magnetic. And we found the answer. I mean, these are macrophages that we know in immunology very well. One of their jobs, of their many jobs, is to degrade old and damaged red blood cells. And in the process of doing so, they accumulate iron, and they build these little nanoparticles inside that. And we found that they have superparamagnetic properties. So at the time when we published that 10 years ago, it was just the explanation of an issue relevant for experimental immunologists. So hardly anyone would be interested in that. But the implication came when I talked to Martin and he said, hey, I have a question for that, because we have been looking for such a long time for the sensor by which birds can sense the magnetic field, but we don't know of any magnetic sensor cells in the body. And then we looked at each other and had this eureka moment. Let's try. That was the story in a nutshell.
In a nutshell, that's wonderful. That's such a cool beginning. So, could you tell me about this experiment? What ultimately was the design and the results?
So, what we do as experimental immunologists, the usual questions that we try to answer is which part of our immune system is responsible for a given function, like, for example, defense against a microbe, or defense against tumors. So you take out this part of the immune system of a model organism. So you take away, let's say, the macrophages of a mouse, and then you ask, is defense against the pathogen still possible? The difficulty with pigeons is that all the tools that we have are rather specific for certain species. So with depleting antibodies or knockout mice that we use, we really can't manipulate pigeons. But the chlorinated liposomes, this is something that is species independent. So it's something we have been using for a long time. Small nanoparticles containing chlorinated, which is a drug that is toxic for certain phagocytic cells that take up stuff. Now, macrophages, the role of macrophages is they are like the vacuum cleaners of the immune system. They sit in all organs, and their job is to remove every debris that should not be there, that could become toxic and could pose a problem in the future. So they gobble it up and remove it. And that's what they do with old erythrocytes. But they also take up these liposomes, and the content of these liposomes is toxic, thereby they die. This sounds a bit dramatic, but it's really not. So the mice and also the pigeons, they can do very well without macrophages for 1 or 2 days. And then the bone marrow has made new macrophages and everything is as before. So we, of course, tested that after treatment with these liposomes, the pigeons are happy and healthy and they can fly. This is one of the controls in our experiments, but they just couldn't navigate anymore.
I got to say, the graphs generated by this experiment showing the pigeons attempting to navigate home are fascinating. I want to focus on that and kind of the experimental release of them and the control conditions. But first, I just had my wife actually was like, Ian, you have to ask, how do you set a homing pigeon's home location, right? And I'm sure it's something to do with, where they roost, where they spend a lot of time. But I'm curious. I know homing pigeons can only, home back to one particular location by default, or I assume. Is that true?
Well, they want to. I mean, you have to make their home as cozy as possible for them. So good food, good mates, and especially, sexual mates, and then they want to return home as soon as possible. So that's good. The way to do the experiment is really to put the pigeons in a situation where they cannot use anything but their magnetic direction sense. And that's actually very difficult because you can train them to fly in a certain direction so that they know, it's always east. But then you have to basically remove the landmarks and you have to remove any information from the sun. So no polarized light, nothing. And to do that, we had to rely upon really, really bad weather. So really low-lying clouds. And usually that's coupled with a little bit of wind and drizzle. So it's conditions where the birds don't like to fly. But then, you have to make the home loft even cozier for them so that they really want to go home, even under bad conditions. So it's actually a really tricky experiment to do in the field. But the key is complete overcast over the entire time. No sunlight and really, really low-lying clouds. And that's what a lot of people don't understand, because otherwise, if you have just a high ceiling cloud, no, that's easy because they have their landmarks. They know how to fly. There are very, very few conditions under which a pigeon needs a magnetic direction. Usually they go by landmarks and by vision under all regular conditions and by, direction from the sun. That's easy. These are the only conditions under which pigeons would rely upon their magnetic direction sense.
So they really use it as a backup mechanism when their other navigational cues of vision and landmarks and familiarity are out of question.
Yes.
Okay. So can you tell me exactly how it was shown that pigeons treated with this clodronate liposomes lose their ability to navigate back home? How many pigeons? How far away? How do you compensate for the lack of familiarity, or how do you induce the lack of familiarity so that they really are forced to rely upon that ability?
Yeah. So you train them to fly home from a westerly direction towards east. So they know, every time I'm transported somewhere, I just have to fly east. And obviously they do remember the landmarks. They know the whole thing. They know they're there, and even after the second displacement, they fly home immediately. And then you have to create conditions where none of the other features are available. So no sunlight, really low-lying clouds, so that all the landmarks are gone. So the clouds are basically above the trees. So you can't see any hills, you can't see any valleys. The whole landscape is completely changed. And then it's really, there's either a cryptochrome system that should guide you, which it didn't in our case, or there are other systems, or if you knock out the macrophages, and then you look at the tracks and you say, well, no, this is very different from what they do if they would rely upon their magnetic sense. They just fly in all kinds of directions. They get lost. I mean, they're flying, but they are not really going anywhere. And that means the most likely explanation is these macrophages are knocked out. They are the ones that are responsible for keeping the direction, and they can't keep a direction anymore.
So just to make sure I'm following, you take a collection of pigeons, you transport them 20-ish kilometers west of their home. Half of them have been treated with this drug that annihilates most of their iron-containing macrophages, which the proposed mechanism is that these are able to form a kind of directional compass and grant the pigeons this ability. And under conditions in which they cannot use the sun, those pigeons treated with the drug fail to fly home. So the ones untreated fly more or less directly east back home, and the others, it's more or less completely random as to which compass direction they fly in from there, and fail to return home within this hour or 70 minutes that the non-treated ones fly home. Is that correct?
Correct. Yes.
Okay, but when it's sunny, when they can navigate using visual cues, then both the untreated and the clodronate treated pigeons are able to fly back home.
Yes. And
okay.
We don't know if they use landmarks or if they use the sun direction, because they have a sun compass. They probably use both, but we haven't separated out those two. They're actually very difficult to separate out.
It sounds very difficult. It sounds difficult to make the real world into a lab condition with the variance of weather and location and all that. Okay, so as I understand it, I tried reading on some of the skeptics after this result was announced, and I'm hoping that you can help me maybe understand, because I'm one of the things that comes up is the fact that, well, perhaps the clodronate, perhaps it's not actually these iron-containing macrophages that are granting this ability, and the drug makes the pigeons unwell or has some other effect on them. But it seems from the data, I mean, the graphs are so binary. It's like 100% success, 100% fail. So it's clearly doing something. Were the pigeons that were treated, were there any observable differences in their behavior, their energy levels, the time it took for them to return home under the sunny conditions? Or was it essentially like invisible as to which had been treated and which hadn't until it was this cloudy sky condition where they had to rely on their ability?
So maybe that's the last thing that I can contribute. And I didn't see any differences in these pigeons. I mean, it's also, you treat them, you then treat them. I mean, you treat them with a drug, and then the next morning you catch them out of the aviary. You put them together in a box. You transport them out there. There's no difference that we saw in these pigeons. So we grab one randomly out of the box and then release it. I mean, read the number and say, oh, this is one of those, or clodronator as a control. Release them. You don't see a difference. I mean, they fly off, and then soon after you see a difference in the real-time data, because some of them, they just take off and go straight towards home. And the others are maybe even leaving in the same direction, but then, they are just veering off because they apparently can't keep a direction. I mean, for us, it was really a mystery or a miracle that, you do this experiment and think, well, that most likely nothing is going to happen. And then
It's
you
so
see,
fascinating.
well, apparently there is some effect.
And what does this seems to me like a blow to some of the other leading hypotheses, namely like the cryptochrome focused one, because I couldn't find anything in the literature about clodronate interfering with anything that should impact the eye, cryptochrome, anything like that. So because this did abolish their ability to navigate, is that the hit that I take it to be against the cryptochrome explanation?
Maybe, but there could be a cryptochrome system and it could be a backup system. But in our case, supposedly they didn't use cryptochrome for whatever reason. I mean, we haven't tested that, but at least it makes it a little less likely that cryptochrome is used in that system. I think it's not a complete blow. It just means, there's maybe something else happening that should be investigated, and maybe one should look at with a sort of a new eye.
But I think for us, I mean, I just want to add, because you asked a question for the clodronate, and I think it's very important to mention that so the clodronate is not specific to liver macrophages, and it is not specific to iron-containing macrophages. So it is a drug, as Christian already explained, that is taken up by phagocytic cells, mainly macrophages. That's why it's so widely used in immunology by people studying macrophage biology. And it is just because of the administration route, the dose, the timing, that we can kind of narrow down the effect of the clodronate towards macrophages that are primarily located in the liver, maybe in the spleen. And, for example, targeting alveolar macrophages, usually clodronate requires a different administration route. But it is not that, we are not claiming that it is specific for iron-containing hepatic macrophages. So I think this is very important, because some people, they ask a lot of questions about it, and I just want to make sure that, it's specific for phagocytic cells, but it's not, specific for iron-containing hepatic macrophages. And this actually brings me to the second question, whether the clodronate would also target the cryptochromes. I mean, as far as I understood, cryptochromes are not phagocytic cells. It's just light-sensitive proteins in the eye, and whenever they are activated by light, then the birds should see some kind of magnetic pattern supervised on the normal vision, and clodronate will not target cryptochromes. So at least for us, since we are not involved in this highly debate, as immunologists, we have this fresh eye on all these theories, and so we depleted the macrophages in the birds, and they needed to rely on Earth's magnetic field, but we didn't touch the cryptochromes. So, apparently, at least to me, it indicates that the cryptochromes in the situation that we put the birds in didn't play the major role or could back up, this failed magnetoreception. Maybe there are several redundant systems, which makes sense in nature. Yeah.
But to your point, if they are redundant, it didn't seem that they relied on that visual redundant one. If it does exist, my lay understanding, it sure seems like a blow to that explanation.
Yeah, but I don't want to get involved in this,
Right.
debate.
Well, speaking of things you might not want to get involved in, this is scientifically a cheeky question. It can only be, a total kind of imaginative exercise. But I'm curious, like you just used the word vision when it came to the cryptochromes. Like the idea is that when the light hits the cryptochromes, it influences the downstream molecular pathways, and that could lead to some sense of vision of the Earth's magnetic field, as I understood it. If you were being put on the spot by a cheeky science communicator to say how you think pigeons would feel this sense of magnetoreception should it come from these iron-containing macrophages, with the understanding that this can only be pure speculation. I'm curious as to what your intuitions say there.
I mean, it's pure speculation, but as we described already in the paper, we see that macrophages and nerve fibers are really, really close. They are lying really close to each other or within the range of 2 to 5 micrometers. And we know from the mammalian systems that macrophages and neurons, nerve fibers, there is a bidirectional communication. So macrophages send signals to the nerves and vice versa. And I mean, they are located in the liver in the pigeons as well, so close. Why shouldn't there also be a communication way? How exactly this is done, I mean, this is future studies that we have to focus on. But for me, at least this close localization is already an indication that this might be a way of how they are communicating. And then via the nerve fibers, I mean, this can, just like every other information that is transmitted to the nerve via the nerves to the brain. So that would be my take on it. But again, it's just speculation, and we have to do some studies to figure this out in detail.
Thank you. No, and the direction of that future work seems really exciting. My question's perhaps even much more speculative, which is, what would your intuition say that is their subjective experience of this sense of magnetoreception in terms of, I can imagine equating it to some kind of, almost like balance, the way that we have our inner ear structure that will hint at,
some kind of gut feeling, kind of
precisely, yeah,
not gut feeling, but liver feeling. Yeah, maybe it is something like that. I mean, gut feeling, it's known that there's a gut-brain axis. There's a strong connection between the gut and the brain, and what people, at least humans, describe this kind of gut feeling, that you cannot, give strict facts why you decide for one thing or the other, but your gut gives you a feeling. I don't know, Christian, what is your take on this?
I think it's completely impossible for us to get a feeling for that. Try to explain to a colorblind person the difference between red and green. They just will not understand. They cannot, but they can have a feeling that these things might look different. But here we have a completely different quality. We have a sensory quality that we just don't know about. I can understand that this is the question that many people will ask themselves, but I think it's beyond our imagination.
I certainly get that. That makes sense. Another speculative question is, it seems that multiple different species and really like lines of evolution have converged on this ability to sense the Earth's magnetic fields, whether that's sharks, birds, turtles. And it's unclear just how widespread this ability is. And I'm wondering, given that many animals have these iron-containing macrophages because they, help recycle the iron from erythrocytes, should we have a completed science of bird magnetoreception, do you think it's plausible that this ability could be engineered into species that didn't innately evolve it in the first place?
And then we first have to find out what exactly makes up this property. So what do you need to have these superparamagnetic nanoparticles in your cells? What do you need for that? If there's a certain enzyme that builds them, certainly genes can be transferred. But will that be enough? I don't know, because after these particles are present in a cell, they also have to function, and they need to activate the cell, and the cell then has to give the right signals to the nerves. So probably it would require quite a bit of construction, and we are far away from being able to do so and from being allowed to do so. Thank God.
Excellent. Excellent. Well, thank you so much for doing this. I feel that there's probably some great questions I've left unasked.
A lot of questions still to be answered. So we have to put up our sleeves and continue our work.
Awesome. Well, thank you for doing it, and thank you for taking the time to share it with us. I really, really appreciate it, and hope you have a great day.
Thanks for having us.
Yeah,
Thank
thanks
you.
for having us, and thanks for the interest.
All the best. Bye-bye.